Welcome back to our class. Like I said in the previous video, we'll be looking at thyroid hormone synthesis in details. And I told us that thyroid hormone synthesis begins from the hypothalamus by the production of TRH in the paraventricular nucleus, and then it moves to the stimulation of the thyrotropes in the pituitary gland, and then it stimulates the release of TSH, which then stimulate thyroid glands to release T3 and T4. So when thyroid stimulating hormone synthesis stimulates the thyroid follicles, the first thing it does is that it actually brings about stimulation, or it activates a cycle. The main aim in the stimulation of thyroid gland upon stimulation by thyroglobulin is to release thyroglobulin. I just want to look at how thyroglobulin is released. Before thyroglobulin is synthesized, the first thing it does is that it stimulates the release of an effector enzyme called adenylate cyclase. Adenylate cyclase is an effector enzyme which converts ATP into cyclic AMP. This cyclic AMP further activates protein kinase A. Protein kinase A now phosphorylates a transcription factor. This transcription factor now stimulates the gene to start making a protein from your messenger RNA. When this protein is being, this messenger RNA stimulate the ribosomes. And what do they translate? The ribosome will start translating, sends these proteins to the Golgi apparatus, and the end product is thyroglobulin. So, in summary, when TSH stimulates thyroid gland, the aim of it is to release thyroglobulin. That's the first aim. It releases thyroglobulin. The main aim is to produce T3 and T4, right? That is the end point. But the starting point is that it wants to produce a protein called thyroglobulin. Is this protein that starts the whole process of T3 and T4 formation in the thyroid gland. So I told you that for thyroglobulin to be produced, Golgi apparatus will be stimulated. The ribosomes will be stimulated, and this happens with the adenylate cyclase enzyme. This adenylate cyclase enzyme stimulates messenger RNA in the ribosome, which then goes and stimulates the Golgi apparatus to produce these proteins, and this protein is called thyroglobulin. So it is this thyroglobulin now that is like the backbone in the production of T3 and T4. So the next step after thyroglobulin is produced is that iodine from the blood has to be trapped from the blood into the thyroid gland cells, the cells of the thyroid gland. Don't forget that the stimulation comes from TSH, which stimulates the thyroid gland to start all this process. And I told you that the first process it does is to produce thyroglobulin. And I've explained how thyroglobulin is being produced. The next thing it does is to trap iodine from the blood into the thyroid gland cells. And so iodine in the blood is iodine. So we want this iodine in a ionized form. So iodine now in the blood is now ionized, because we want it in an ionized form. And it has to be ionized first. So when it comes into the thyroid cells, the concentration is very low. So we need higher concentration of iodide in the thyroid cells, so that this iodide can now go and bind with thyroglobulin. So how do we bring in more iodide? Don't forget that it's the iodized form. That is the iodide. The iodine is the one we know, the one we take in as food. Then the iodide is the ionized form. And so iodide now, which is the iodized form, we want it in higher concentration. It's very low inside the thyroid gland cells, but it's higher outside in the blood. So sodium, it's outside. Sodium is going to help us transport this iodide into the cells. Sodium is actually low outside and is higher inside. Sodium is higher outside, lower inside. So sodium is going to move from a higher concentration to a lower concentration. I told you that sodium is lower inside and higher outside. So sodium is going to move from a high concentration to a low concentration. So sodium now helps move this iodide, and it does this by using a co-transporter called a sodium iodide symporter. This sodium iodide symporter helps move iodide into the cells. It doesn't really need it doesn't use ATP for this process, and so it's a secondary active transport. Now, iodide now is inside the cell. Iodide inside the cell now needs to be moved inside to the lumen of the thyroid gland. So the iodide inside the cell needs to be moved into the lumen of the thyroid gland. So this can be achieved by an enzyme called pendrin. Pendrin helps push this iodide into the lumen of the thyroid cells. Inside the lumen now, there is an enzyme that will help kick off the iodide and knock off some of the electron and attach it to the thyroid globulin molecule. This enzyme is called thyroid peroxidase enzyme, TPO. This thyroid peroxidase enzyme kicks off some electrons in the iodide, thereby oxidizing it and then attaches it to the thyroid globulin molecule.